Good day, Doctor. My FYP title is the physicochemical comparison between virgin coconut oil and virgin coconut emulsion. So, virgin coconut oil is well known for its health benefits such as antioxidant, antimicrobial, and antiviral, which is very famous for VCO. So, on the other hand, what is VCE? VCE is when virgin coconut oil and water, which are two phases that usually doesn't mix together, and with additional of surfactants such as lecithin, it will mix to become small tiny droplets of VCO, which is an oil and water emulsion called VCE. So this one is the virgin coconut oil, and this one is the virgin coconut emulsion made. So here are some important literature review throughout my thesis, which is the first one that compare the physicochemical properties of commercial VCE. Second one, they hydrolyze VCO into the monoglyceride and test their antibacterial properties. And last one is the recently done in silico research of VCO components doctored with 9 proteins of SARS-CoV-2. So in the methodology, this is my formulation of my VCE. So first is to activate the lecithin with water and mix it for about 10 to 15 minutes. And when it is activated, add the pre-measured amount of VCO droplet by droplet. And then is the adding the water phase, which consists of water, xanthan gum, and glycerol, and then homogenize the mixture for about 1.5 minutes, and I will obtain the VCE, which is an oil and water emulsion. So next is the physicochemical test. First one is the solubility test in water, mixing the VCO and VCE in water, and then food coloring is dropped into the VCE or VCO, and then also a pinch of curcumin powder or turmeric powder, which is the subtitles, the local name, into the surface of VCO and VCE to determine it dissolve or not. So then is the Osiris and Molinspiration software to calculate the physical chemical properties of the VCE, VCO components, and some reference drugs to find the drug ligands and bioavailability. The left side is the Osiris software and the right side is Molinspiration. So for the molecular docking, the Autodoc Vina 4.2, the latest version was used. So this is in the Autodoc software viewing the aureus DNA binding protein. PDB ID 4PQ, which was used. The green one is the ligand, which is in this case is monolaurin, doctored with the protein of aureus. And then next, the antiviral test is using the SARS-CoV-2's main protease, which is obtained from protein data bank also. The code is 6LU7. So the green one is monolaurin, which is an example of the VCE component doctored. And for this antiviral test, all the VCO and VCE components are doctored twice because this is a rather new research that. So I want to get a more significant and more accurate result. So the docking was doctored twice. So moving on is the visualization of the result of the molecular docking. For 2D, I use the protein plus, and for 3D, I use the protein ligand interaction profiler. Then finally, pyramid, and now is the result of my FYP, and which is firstly solubility test in water. So this one is before mixing the VCE and VCO. VCE is clearly showing solubility already in water, and VCO is clearly not soluble. It just float on the water. And then next is VCE. When VCO have mixed a little bit, VCE is clearly seen it totally dissolved in water, and VCO didn't dissolve at all, but it forms a micellus on the top of water. And then is the food colourant in VCO and VCE, where VCO did not allow the food colourant to dissolve, and when the food colourant is dropped into VCE, it starts to dissolve slowly, as seen here. And this it allows the food colourant to dissolve because the VCE has already been polymerized or emulsified by the surfactant lecithin, forming a layer around the VCO into small droplets, and the polar has allow interaction of water and any water-soluble stuff, allowing them to be VCE to be dissolved in water. So as a control, water was also used to test the food colorant, and it shows total dissolution inside water. So the solubility of turmeric powder in VCO, VCE, first VCO and a pinch of turmeric powder, it totally dissolved in VCO. And for VCE, it didn't dissolve and it just remained on the surface after a few swirls. It's still the same, it doesn't dissolve. And as a control, the powder was also put inside water and it just forms a turbid mixture. It means that the turmeric powder cannot dissolve in water. And overall, all the solubility tests has proven that the VCE was successfully formed and no water, which is an oil in water emulsion, and no oil phase was shown because it does not allow the turmeric powder to be dissolved here, as seen, and it dissolved in water just now. So for the Osiris and Molinspiration calculation, here are the results, and VCO and VCO, the VCO components did not show very good Osiris drug score based on their physicochemical properties, but they show moderate Molinspiration score, and all the VCE components show good Osiris and Molinspiration score. For you are wondering why all the component used for VCE was the monoglyceride, because when VCE is emulsified, when it enters when in our body, when we drink the VCE, it doesn't require bile acid to be emulsified again, so it already is readily to be broken down by the lipase to form its monoglyceride form of the VCO component inside the VCE. So that's why all the component used for VCE was the monoglyceride component instead of the fatty acid components like VCO. This also same for the autodoc later. So the lecithin also did not show very good result because of the long hydrophobic tail. These are all the lecithin components, but only when in the VCE form, only the polar heads are interacting with the water surface, and it is broken in the body easily when by lipase later. So here are some reference drugs result from the Osiris and Molinspiration correlation. But Some show very good results, such as amoxicillin and penicillin for antibacterial results, for the antibacterial drugs, for fluconazole as an antifungal drug, but not for remdesivir and ivermectin, which are antiviral drugs candidates for SARS-CoV-2. So moving on is the molecular docking result for aureus DNA binding protein for the antibacterial test. These are all the best few components, best hits of the docking. So from VCO is capric acid, and monolaurin from VCE, and the reference drug penicillin obtained the best results. So the green lines are hydrophobic interactions, and the dotted lines are hydrogen bonding. So this is a 3D visualization of the docking with the amino acid components, which will be shown further. Okay, so this is for the best docking for monolaurin with the antibacterial, so for the bacteria protein. The blue lines are the hydrogen bonds, and the dotted lines are the hydrophobic interactions with the amino acid residue shown here. So next is the antiviral docking result for SARS-CoV-2 and Pro6LU7. These are the best results with linoleic acid from VCO and monolaurin from VCE, and ivermectin from the reference drugs obtained the best docking hits these few components. So this is the 3D view, and all of them bind and is targeted at the hydrophobic pocket here of the protein. So this is the interaction of the ligand monolaurin, the best result from VCE with the amino acid of the main protease of SARS-CoV-2, which is the blue lines are also hydrogen bonds, and the dotted lines are hydrophobic interaction. And for this interaction, there is a salt bridge, which are the yellow dotted lines which connects, which is also another significant interaction that allows strong binding of the ligand with the receptor, which is good. So next, and finally, as a conclusion, VCE is superior in many aspects in terms of physicochemical compared to VCO, where VCE have a better solubility, and the monoglyceride in VCE, which is not present in VCO, have better physicochemical property proven by the Osiris and Molinspiration calculation, and overall monolaurin from the autodocking showed the best results from VCE compared to VCO. And with these two results, with the 4PQ and 6LU7 protein, which it can be a good antiviral or antibacterial drug candidates, if further studies and further research are done. So for the limitation and recommendation, boiled water was used instead of distilled water, and hand homogenizer was a normal one bought online, was used for the formulation of VCE, and I prefer one is distilled water and lab homogenizer, and also to determine the components of VCE and VCO, GCMS is recommended in future studies, and to further understand the solubility and absorption of the VCO and VCE in the human body, PBPK modeling can be done, and also for the in silico modeling, which is only preliminary studies only, In vitro, which means a lab conducted research, is more recommended. And also other physicochemical properties can also be done, which are not done in this study, such as zeta potential, rheology, and even the microscopic view of the VCA and VCO. And that's all from me. Thank you for now, and have a nice day.